So Dr. Medicine and postdoctoral fellow at the Institute of Physiology at the Johann Gutenberg University of Mainz in Germany. He's done some fascinating work exploring the role of entropy on boredom that I've been very interested in myself. And if you haven't read it yet, there's also a great piece showing computationally how boredom might influence trend setting. And I highly recommend that article. It's a great fun read. Today, he's going to talk to us about how low sensory information affects cortical activation and drives boredom-related behavior in humans and mice. So I'm really looking forward to hearing from that. Johan, if you want to start your screen and get started.
Thank you very much for the kind introduction, James. Perfect. You should all see my screen. Work?
Yes.
Okay, nice. Yeah, first of all, I would also like to thank the organizers for setting up this delightful online conference. So it's a great pleasure for me to talk here and to present to you some of our research that we conducted over the past years, exploring the neuronal and cognitive underpinnings of boredom. But let me start with spending a few words on what still fascinates me about boredom. So I feel it's very astonishing that boredom is so ubiquitous, right? And as humans, we all know this state and we experience it almost every day. And I showed this painting from Renoir because I think it nicely illustrates one situation in which one might encounter boredom. But of course, there are various more and various other situations where boredom hits us. And so boredom is not only ubiquitous, it is also multifaceted in respect to the conditions that elicit this state. So this is also reflected by a wide range of psychological definitions of boredom, characterizing it as a consequence of, for instance, lacking attention, a lack of meaning, lacking agency, or also broadly as a mismatch of an individual's cognitive resources and the current demands of a situation. So these definitions are all based on solid empirical work, and they all are true in some way, right? However, they highlight that boredom can result from different causes that, however, converge in one shared experience that I think is dominantly characterized by negative effects. So this is a key feature that not only impacts how we perceive boredom, but also it holds a function for boredom by inducing behavioral changes in order to alleviate or mitigate the causes of boredom. So according to this perspective, boredom is a driving force of behavior or cognition that prevents individuals from becoming stuck in monotony and fosters exploration of one's environment. And of course, you might imagine that such a driving force is highly essential for human behavior in order to continually learn, but also for non-humans, it might hold evolutionary advantage. And indeed, looking at the literature, there are studies conducted with mink, for example, or also with rodents like mice or rats that show boredom-like behaviors. So it's really a phenomenon that does not only concern us humans. But now you might ask, where in our real life do we find boredom? And here, coming from a clinical background, I'm still amazed by the wide variety of clinical conditions that have been associated with boredom, including neuropsychiatric disorders such as ADHD or depression, but also traumatic brain insults, so structural brain lesions are associated with elevated and chronic boredom experience. And on the other side, there are also various impulse control deficits linked to higher boredom experience. So it really is a phenomenon with high importance. And during my time working at the psychiatric department, we aim to explore this a bit more, investigating the role of boredom for the treatment outcome of psychiatric patients. So what we did in the study is that we screened all patients that were admitted to our clinic over a course of 3 months for their state boredom upon admission and then 1 week later. And comparing the patient data to a healthy control cohort, what we observed is that all different types of psychiatric disorders that we tested, so psychotic disorders, addictive disorders, affective disorders, etc., they all showed elevated levels of boredom that persisted even 1 week after therapy initiation. And boredom was not only elevated in these patients, it was also predictive of their outcome measured as the treatment duration of these patients. So patients that were more strongly bored also stayed in the hospital for a longer time period, indicating that boredom here could serve as a complicating factor in psychiatric treatment. So with this, boredom is ubiquitous, right? It is of high psychosocial importance, but still many questions about the nature of boredom remain unresolved. And one question which I would like to discuss with you today, which I think is very central, is the question about the neuronal and cognitive underpinnings of boredom. Because I think these mechanistic aspects of boredom very much limit our current understanding of this phenomenon. And in our lab, we address this question in two main branches of the research. On the one side, we conduct experiments with humans where we want to make a link between different behaviors and boredom experience. Also, this branch allows to conduct clinical trials. But on the other side, we also do experiments with animals, and we use mouse subjects in our lab where we have a wide access to different experimental tools to assess brain activity in order to dig into the neuronal underpinnings of boredom. Right? So in my talk today, I will talk about results from these two branches. And first, I will describe to you a behavioral paradigm that we established in order to assess boredom across different species. Second, I will talk to you about the drivers of behavior in these tasks that are also linked with boredom. And in the third part, I will talk a bit about the neuronal underpinnings of boredom. So let's start with the first section. When asking ourselves the question of how to assess boredom or how to construct a task in order to measure boredom, we decided to operationalize the key feature of boredom experience, which is, as I mentioned earlier, negative affect. And as boredom is often induced by monotonous stimulation, we wanted to put participants, in this case it was human participants, in a choice situation between monotony and a less monotonous variable alternative and measure boredom in the form of monotony avoidance. So we had a modified two alternative forced choice task where one alternative, so one button on the screen, was associated with a single sensory stimulus, whereas the other variable alternative was associated with multiple different stimuli compiled in a larger stimulus library. And in this task, whenever subjects made a click on the left monotonous alternative here, they were presented with a repeating image of this shoe, whereas whenever they chose the right alternative, they were presented with varying images. So this task allowed us to measure the preference of individuals between monotony and variability. And we not only compared monotony to variability, we also had control conditions where both alternatives were equivalent. And this task we designed not only in with visual stimuli, but also in an auditory version where we used recordings of spoken German words as stimuli. So the data I will show you comes from around 100 healthy human participants. And let's look at the choice data. So as expected in the control conditions where both alternatives are equivalent, if we look at the choice probability over the time of the task in visual modality here in blue and in auditory modality, we see that choice probability remains at chance level, indicating that there is no significant bias towards any of the sides. However, interestingly, when now comparing the variable versus monotonous condition, we find that subjects here learn to discriminate both alternatives, starting to avoid the monotonous option until reaching a plateau preference of about 80% of the time choosing the non-monotonous option. So across these two modalities, humans in this task show a robust avoidance of monotonous sensory stimuli. Now the question, of course, was if this behavior has to do anything with boredom, and therefore we let participants rate state boredom on a visual analog scale before and after the task. And by comparing these two measurements, we found that each single participant rated higher boredom after the task, indicating that this paradigm indeed was pretty dull and that we induced a high degree of boredom. And to be more specific, we then also quantified the pooled behavioral bias across both modalities for each participant and correlated it with self-report gold standard measures for state boredom, namely assessments of the multidimensional state boredom scale. And here we found a significant positive correlation, right? Indicating that subjects with higher boredom also tended to show a larger monotony avoidance. Well, this was all humans, but I told you we are also working in mice. So in the next step, we wanted to translate this paradigm to animals. And therefore, we used a slightly modified version of the task where we measured monotony avoidance as place preference. So mice here were put into a Skinner box that was separated into 2 zones. And mice were able to freely move between these zones and were tracked whenever they were located on one side. They were presented with monotonous auditory stimuli, whereas the other side led to the presentation of variable auditory stimuli. Here you can see the example movement trajectory of a mouse, but when then quantifying the place preference over multiple mice and multiple sessions, we found a picture that was very similar to what we observed in humans. So in control conditions with equivalent zones, mice did not show any systematic bias, whereas when comparing monotonous versus variable stimulation, mice started to avoid monotony, showing a monotony avoidance bias that built up over time very much to what we find in humans. Okay, so with this, we had a behavioral proxy for boredom, or at least a signature of boredom-related behavior. And in the next step, we wanted to dig a bit deeper and look at the real situational drivers of this behavior in our task. And for this, we again conducted a human replication experiment where we now not only measured choice preference at extreme degrees of monotony, but also at a spectrum. And for this, we started with this condition having 1 versus 1 stimulus, and then gradually increased the amount of stimuli at one alternative until coming up with 1 versus many stimuli, which would correspond to monotonous versus variable. Then we just closed the circle by making the other alternative also more variable. And in each of these task conditions, we assessed the preference for one reference alternative, allowing us to look at preference over different ratios of stimulus library sizes. And first, what I show you here is a replication of our previous findings, indicating that when both alternatives are providing the same type of information, we do not have a bias. And when comparing monotony versus variability, we found that monotony is avoided. Now, the space in between looked kind of surprising for us because this preference curve was neither linear nor symmetrical in any way. And this becomes even more obvious when overlaying those 2 sides of the curve. So initially, we thought that the ratio of library sizes might be a good descriptor of task experience. But if you look, for instance, at this condition here, where we have 8 versus 1 stimuli against 64 versus 8 stimuli, having the same ratio, however, showing highly different degrees of choice preference, we see that this is not a good descriptor at all. So this led to the question that I discussed with my dear colleague Ohad, who is now working at Yale University, of how to quantify the really experienced degree of monotony of subjects in the task. And for this, we came up with a measure from information theory, namely the empirical entropy, which is a concept developed by Claude Shannon that describes the information content or the variance in a distribution of events. And this term can be used antagonistically to the term monotony in our task. So in our task, we then specifically quantified the amount of information, the amount of entropy that was provided by both alternatives at a given point in the tasks. And for instance, one alternative that previously led to the presentation of a repeating stimulus would have a low entropy, whereas an alternative that led to the presentation of multiple different stimuli would have a high entropy. Then we built the difference between these entropy scores for both alternatives and looked in how far it predicted the upcoming choice. And what we found was a curve that we found very interesting because we observed that choice behavior directly depended on entropy difference, where higher entropy for one alternative predicted that it was more likely that this alternative was chosen in the next trial again. And when using the same framework and analyzing our mouse data with it, we found a very similar relationship, suggesting that this boredom-related choice behavior that we observe in this task is directly driven by states or experience of low information content. So these findings suggest that information plays a critical role in the computation of boredom. And in the last set of experiments, we assessed the neuronal underpinnings of information in the brain. And for this, we conducted, again, an experiment now with mouse subjects, and we used a methodology to record brain activity, which is called calcium imaging. So this calcium imaging basically depends on an injection of a viral vector that after some incubation period leads to the expression of a certain protein in the mouse brain, namely a protein that serves as a calcium indicator. So this calcium indicator always provides a fluorescent signal whenever there is a calcium transient in one cell, and as calcium serves as a proxy for neuronal activity, when we then take our mice and put them under the microscope, we're able to record from hundreds of neurons simultaneously while having a living and awake animal that we can present with different sensory stimuli. So it's a quite powerful technique, I would say. And in order to bring in now the entropy or the information content, we presented our animals with stimulus sequences, so sound sequences that systematically varied in their variability. So we had some sequences that were entirely monotonous and others that were entirely variable. So here each dot represents one sound, and the color of each dot represents a different sound identity. And by using the information theoretic framework, we were just able to compute the information content for each single instantiation of each sound. Here you can see an example snapshot of this microscopic recording, where here each red dot would correspond to the nucleus of a nerve cell, the neuron, whereas the greenish signal reflects this calcium signal that, if you follow this green signal over time, allows to compute or construct these response traces for hundreds of neurons simultaneously. Okay, so the advantage of our paradigm here, using these sound sequences, was that we could really test the encoding of information content with controlling for other types of encoding stimulus-specific features. So we took all instantiations of one particular stimulus, for example, this bluish sound here, and in our set of sequences, we had instantiations where the sound occurred where it was well predicted by the previous stimuli, therefore carrying only a low entropy. And we also had instantiations of this bluish sound where it was not predicted by the prior stimuli, therefore carrying a high entropy. And with all the instantiations that we had across our sequences, we were able to nicely cover a broad spectrum of entropy conditions for a given stimulus. And comparing the neuronal responses over entropy, we found two main things that I will describe to you. So first, we observed that information content is quantitatively encoded in neuronal activity, where stimuli with higher information also evoke larger neuronal responses. And on the other side, we also observed that information is encoded by employing specific activity patterns in the brain. So we found nerve cells that increase their firing rate with increasing entropy, and some other specific subsets of neurons that decreased their response rates with entropy. So indicating that there are distinct neuronal ensembles specifically tuned to information content. So tuning here reflects that the cell in its activity pattern is responsive to this feature of entropy or of information. With this, let me summarize. So first, I showed you a behavioral paradigm that we established where we can assess boredom in the form of monotony avoidance. In this paradigm, we find that boredom-related behavior is directly driven by states of low information. And lastly, we find that information content in the neocortex is highly represented and in two main features. The one side, we have encoding in the response magnitude, and secondly, we have an encoding of entropy in the specific patterns of neuronal activity. So with this, I'd like to close the circle, go back to our initial Renoir painting and complement this by another painting from Renoir. And concluding from all our findings, I think boredom can very well compare to a cognitive signal such as hunger. So hunger is a state arising from low nutrition, indicating to a subject that it should get or seek for food in its environment, whereas boredom is also a similar aversive state, however, arising now from a state of low information and signaling to an individual that it should seek for any information source in its environment. And in order to allow the computation of boredom, I think it's essential that brains not only encode what types of stimuli are present in our surrounding world, but also how informative these stimuli are for our internal models of this world and how surprising they are in order to effectively guide behavior and optimize information seeking. So if you're interested to read a bit more about this perspective, we recently published a short review on this issue together with my colleague Ohad Dan. And this being said, there's nothing left than thank all my collaborators. In particular, I would like to thank my advisor, Simon Rumpel, who I really enjoy working with. And I think this is a very fruitful time. So I'm really thankful for his mentorship. And also, I want to highlight the contributions from Ohad Dan, working at Yale University, with whom I developed these information theoretic ideas. And thank you all for listening. I'm happy to take any questions.
Fantastic, Johan. Thank you very much for that talk. You can see the applause emojis and everything coming from there. It was really dense and a wonderful wonderful presentation. As the moderator, I probably should let other people ask questions. And I see that Wynand has his hand raised, but I'm going to beat you to the punch there, Wynand. I've got about 10 questions I want to ask, and if I don't get one in, then I'll feel bad about it. So I want to ask about the other end of the entropy scale, right? So you've just sort of told us that when I've got low entropy and everything's monotonous, that I'm going to be bored. What about the upper end of that entropy scale where things are highly chaotic and random?
You're entirely right? So one might expect a U-shaped relationship of boredom over different degrees of entropy. So if everything becomes too complex, boredom should also arise. And this is something we do not find in our behavioral task results, because still, like just looking at how participants describe the task, even if it provides variable sensory stimuli, it's still pretty repetitive, right? It's not that participants would go out of this task and say, well, it's super interesting, like all these different images of everyday. So I think just with our task, we do not capture this full spectrum. However, you're right. Yeah.
Well, I was going to sort of follow up with that just quickly, because I do see other hands. And it was also interesting to me on your very first data slide that people only chose the variable thing at about a rate of 80%. So, so why are humans still going to the monotony towards the late end of your task? And mice seem to be less neophilic. The mice didn't seem to have the same rate of choosing the variable stimuli. What's your thoughts on that?
So I think that there is also maybe some motor entropy, if one might call it like this. So just trying out different motor programs and also exploring in a way different behaviors and different options that this task allows is something which can also generate some information that is independent from the sensory stimuli that the subjects experience during the task. So I think this is on the human side. And regarding the mouse comparison, so mice in this place preference task show a slightly different behavior. So they do not constantly switch between zones. However, after some time, they just settle in one of the zones. And what I showed you was like the average probability over multiple sessions. And this, their mice showed a higher probability to settle in the less monotonous. So.
Excellent. We'll go Wynand and then Vanya and Sarah.
Right. Well, thank you for a fantastic presentation. Really interesting. I also really am fascinated by this link between boredom and entropy. So thanks for that. Also, impressive to see that you've extended the slightly sadistic tendency of experimental psychologists like us to bore people to actually bore other animals too. So very impressive. So I have a question about you, and I have no idea how it can be answered or how I would answer it either. But so you explain boredom, you account for boredom as something that's arising from a lack of information, or at least that people who are bored can come at that potentially with greater variety in information. Do you think it's possible to distinguish, either with your paradigm or modification to it, between boredom being related specifically to information acquisition or to attentional engagement? Are these two different at all, or are they effectively identical, but we're just using different words for them? So I'm not sure, right? So the question is basically, are you explaining things using the acquisition of information as an important variable, or is it about attentional engagement, or are they just the same thing?
Yeah, thank you very much for this question. I think it's a great question, and this concept of information in per se is pretty complex, right? So here we estimate information content, or in the previous study I showed you, we estimated information content with empirical entropy. However, the truly conveyed amount of information does not only depend on what is sent out by a certain sensory event, it also depends on how well this information which is sent out can be decoded and received by an individual. And here I think, as you said, attentional mechanisms play a crucial role, and it's really difficult to disentangle these mechanisms. So in a recent study, we looked at different types of information perception using textual stimuli. In a human study, maybe there we go a bit into this issue. So this was published in Communications Psychology this year, if you're interested to read more, but definitely both plays a role. So on the one side, the variability of a message which is sent out, and then on the other side, also an individual's capability to interpret and decode this message.
That's interesting.
Vanya.
Yeah, so yeah, I agree. Thanks a lot. That was super interesting. And I think I will just have one question, and then I will send you an email and invite you to our lab meeting so we can discuss more. I think that's more efficient. And so
I'd be happy.
It'll be fun. So I really do that. So my question, or the question I want to ask now, is more coming from my work that is also related on effort and boredom and kind of how those two kind of are related. And I was just wondering in the mice paradigm, because if I understood correctly, in the box, they basically move to an area where they get a stimulus or not. And typically, there's a lot of old research also showing that non-human animals minimize energy expenditure whenever possible. And I was wondering if you could, or if you did, kind of track the amount of energy consumed in terms of steps taken, in terms of forgetting or avoiding certain state of information, which would kind of be an interesting way to kind of see how people trade off the cost of monotony versus the cost of additional energy expenditure, which would be, that is super interesting for me. Have you any insights on that?
So we didn't test it explicitly. Also, again, thanks for this wonderful question. And I would wish to be able to say more well-founded, data-driven things about it. But everything I can do is speculate. And I mean, mice show this tendency to not only be energy efficient, they also explore. So they show inherent information-seeking behaviors. And well, it's a debate in how far information-seeking behaviors are driven on the one side by boredom, and on the other side also are driven by curiosity, right? So to just fill information gaps in what they know about their world. And I think both plays a role. And it would be interesting to kind of trade off sensory information with metabolic energy. But so far, I mean, we don't have any particular grasp on this in an experimental context.
Okay, we'll go with one last question from Sarah.
Yeah, I can only repeat what the others said. Thank you. It was super fantastic to listen to you. I come from the animal boredom world, and I was wondering about the paradigm you use, because I really like this with the place preference in the mice. And I was wondering, you sometimes talked about boredom assessment also in your conclusion. And I was wondering, isn't it more boredom induction somehow, eliciting boredom? So we always need to be super careful because we don't have, we cannot ask our animals how they feel, you know, so we need to be careful to avoid circular reasoning. And I was wondering, can you say something about it very briefly? Thank you.
Yeah, thank you for this question. Let me go back to this slide describing our task. Okay, so that's a very good question. And I think in our task, we find both components. So on the one side, this task induces boredom. On the other side, we also provide subjects with the opportunity to be active and at least mitigate boredom a bit, right? So this variable alternative still is not very interesting, but it is less boring than the monotonous one. And this is the choice situation that we give to our humans and mice as well. And regarding the second part of your question, can we be sure if we are measuring boredom in animals? Well, as you said, right, we cannot ask them. We can just say that we observe a similar behavior to what we find in humans. And there we find this correlation to self-reported boredom. And also in mice, we tested the effect of information content on pupil size and find that there also low information is associated with lower pupil size, so lower levels of arousal, which would also accord with some classical definitions of boredom. So there are at least some hints, but I mean, to answer it with full certainty, I think we need more data.
Excellent. So, Johan, if you can stop sharing your screen and Wynand, you could start. Johan, you'll see a comment from Josefa in the meeting chat, so you might want to just try and have a read of that and respond to her question.